Put your hands together for Carlo Ventura. Louder, guys, come on. That's better.
Well, thank you so much, and deeply touched. This is the first time for me in this conference series, and I would like to thank the organizers and Richard for giving me the opportunity to share with you some of our efforts. So the title is maybe quite provocative, Voices from Stem Cells. So can these cells talk? Can they emit a sound? Yes. There are mutant vibrations all over around the body. We already heard about this in the previous lecture. There are mutant vibrations in the DNA. There are mechanical, electromagnetic vibrations. One can influence the other. Mutant. Whenever a system goes into adaptation, it's going to die. That's an important message. Music relies upon avoiding continuous adaptation, changing the rhythm. It's very important. Our cells, they continuously change their rhythms. They rely upon avoiding adaptation and changing the pause interval. So absence of vibration is also important time to time. And this is a new paradigm for tissue regeneration. The lab that we set up just 3 years ago, and my friend Luc Montagnier he inaugurated this new lab. It's called SWEET, that means Stem Wave Institute for Tissue Healing. That means that self-healing may rely upon a signaling wave, and then we'll try to. Do you want to stand here? Okay, sorry. Okay, here is. You can see everything the screen. Okay, fantastic. Too much technology, so then at the end of this talk, I will tell you what these symbols are. There is a National Institute for Biostructures and Biosystems involved, which is also partner of the NIH and involves 23 Italian universities. And I will say very importantly, there is an Arts and Science Institute, because most of the experiments that you will see have been designed together by artists and scientists. We believe that arts is a major opportunity for human being for develop our consciousness, and that any form of art can talk to the innermost dynamics of our biology. So vid art science. Vid comes from the Latin, imperative Latin vide, have a vision. Have the courage to, the brave heart to change a paradigm. So hope it works. It works. So the lecture is going to be initially focused on the finding that our cell, every cell, including the stem cells, can produce and sense electromagnetic fields, mechanical vibrations. That also means acoustic vibration and light. And that we can use these physical energies to afford major changes in the regenerative potential of our stem cells. Every tissue of the body has its own resident stem cells. So we talk about tissue resident stem cells. You have to conceive since from the beginning that from the scratch on our skin up to the fracture of the bone or hepatitis, whatever we experience in our life, that we are targeted for self-healing. So sometimes the threshold for self-healing increases, and this is what we may call a major disease. Another fundamental issue is aging. It's well known that a newborn will repair any tissue damage very easily compared to an elderly person. This is also because our tissue-resident stem cells do age while we are aging. And I'll show you that we can use this physical energy in such a way not to prevent, but to reverse stem cell aging. So a molecular rejuvenation of the stem cells that we have inside. A seminal discovery within these fields already came in 1992. This genius, Günther Albert Buehler, made a very nice and provocative experiment, and the paper was published in a very nice journal, PNAS. So what he did, he grew cells very crowded on the surface of the thin glass layer, and then another cell populate, the same kind of cells were grown at a very rare number underneath. So crowded cells, rare cells. And we are in the dark of the incubator. And by dark microscopy analysis, then he found that the cells, the rare cells, were always aligning themselves at a given angle with the more crowded cells. So 0 to 30, 30 to 60, 60 to 90 degrees. So we are in the dark in the incubator. And these cells can recognize each other through the glass layer and orient themselves at a precise angle. To prove that this was a specific phenomenon was made by the fact that he deposited very thin, only 20 to 100 angstrom of nickel chrome layer, which absorbs light in a very wide spectrum, as you can see here, and the phenomenon disappeared. While the same thickness of a latex layer, which only absorbs light from, let's say, 700 nanometers to 400 nanometers, leaving passages through infrared and near-infrared light, would allow the phenomenon to be preserved. So he concluded that cells are able to detect the orientation of each other by signals that penetrated the glass, but not a thin metallic film. And these signals are carried out by electromagnetic radiation, right? Light is a form of electromagnetic radiation. So this is a very conventional journal, very important one. Many Nobel Prizes have published their main discoveries in this journal, or Nature or Science. Later on in 2005, he added some new concept to this discovery. And he demonstrated that the fact that this near-infrared light was scattered inside the cells was a major mechanism for cells to recognize each other. So scattering of light is, it's like I have a beam, light beam that goes to an empty room. It may have a straight trajectory, but if it has to go through all these people, it will jump from one person to another. So this is kind of intuitive meaning to talk about light scattering. And he found that there are centers for light scattering, like around the nucleus, the peroxisomes and the lysosomes. He could load cells with particles of different diameters, latex, or diamond, dark and white diamond, just to increase the intensity of light scattering. And it could provide evidence that the cells were aggregating even more. So the first form of inter- and intracellular recognition is migration and aggregation, when the cells create a network. This is already continuously going on in our body. He measured the intensity of light scattering. We're still in the dark of the incubator, so we are not sending light to the cells. This is light produced by the cell. It was the first clear-cut evidence for a communication of this type among cells. The number of aggregates and the interaggregate distance were related to the intensity of light scattering. Now, this is a movie from Günther Works, and here he is sending light to the cell, just to show to the audience what's going on. As the cell receives lights, the cell moves around an obstacle with no random movement, but with a specific trajectory. You can feel the obstacle. It's not random search for the obstacle. It goes just around with a specific trajectory. And so, movement. what happens inside the cells. These are pictures taken by confocal microscopy with a technique that allows us to visualize inside human fibroblasts, the cytoskeleton. Inside the cells we have microtubuli, and these microtubuli are continuously vibrating, moving. Let's see. This is a plasma membrane. The surface of the cell, it's like wings moving. Here we have receptors for substances, for whatever. Can you see? It's always vibrating. Then we have to understand what's the meaning of this vibration. Just have a look at the cell, the cytoplasm, the nucleus. Here you have DNA. And you can see the microtubuli are continuously vibrating, but they cross continuously with a specific code from the cytosol to the nucleus and vice versa. Now the signaling molecules do not swim into water cells. We are made of water at a high amount, 77%, even more, but the molecules do not swim because that water is non-homogeneous gel form. So the molecules, I'll show you later, they walk and they dance through these physically oscillating cables. Here inside you have 2 meters of DNA, 2 meters and 10 centimeters. Can you imagine? And this diameter is 50 to 80 microns. Each loop that DNA makes is important to have a transcriptional machinery capable to tell our genes how they should behave, not as a single entity, but as an ensemble. And you see the network of this cable, this microtubuli, is intrinsically vibrating. You can actually see that something is going out, some tubuli are crossing the nuclear membrane through the nuclear pores in the other direction, inside, outside. There is no first inside and then inside or vice versa. It's a continuous crosstalk. So the DNA is vibrating. The scaffold here is vibrating. You see that the density of this network is also changing. It's more intense or more rare. If I look at the dynamics at a closer look inside the nucleus of each microtubuli, you see this sort of dance. We call it buckling. And they do not move because they move like this. They proliferate at one end and they are demolished at the other end. By convention, we call it the plus end, the end that polymerizes, that grows, and the minus end, the end that is demolished. So the movement comes from this growing at the one end and being demolished at the other end. But we also have a physicochemical plus and minus end. The growing end is rich of histidine, lysine, arginine. So these are N-terminal rich amino acids, so the positive charge. While the demolishing end is rich in aspartate and glutamate, carboxylic group with a negative charge on the oxygen. So you have a physically oscillating dipole. And so it's a clear example where a mechanical vibration also generates electric and magnetic field. And this field has been first mathematically demonstrated, but then this fantastic work by the group of Anirban Mandiopaidai provided direct measurement of magnetic fields and signature for magnetic fields even from a single microtubulus. So this seminal discovery involved complex technologies. So I will go fast in this part. If you have questions, specific questions, I will be happy to answer. But basically, Anirban has been using 2 techniques. Scanning tunneling microscopy, STM, which applies a voltage to a very, very thin tip, which is able also, like to play with the surface electron of the matter, and when the electron from this excited state, they go back to the ground state, they emit energy, but in such a way that you can visualize the form of the structure that is producing this change in voltage. And this is tubulin. Alpha and beta tubulin are the building blocks for these nanowires that we call microtubuli. And you can see here that probing with STM microscopy, a tubulin molecule has a voltage or a change in conductivity whose profile is totally different from the profile that you can get by probing the microtubules. So assembling the building blocks in this structure makes the structure behaving as a conductive system with a sharp image at minus 20 millivolts, totally different. What he did then, he created a nanochip. He grew in vitro this microtubulus on a chip of platinum electrode to create a sort of nano sandwich. Then he used one part of this chip as an end of a circuit, and the other part was the tip of an atomic force microscope, which is the atomic force AFM. It's like a small arm, let's say 100 micron, with a very thin probe at one hand that we call carbon nanotube. It's a tube in 5 nanometers diameter, only made by atoms, carbon atoms, ending with a single atom. That's like having your tip of the finger ending with a single atom and touching the surface. You can move your finger like a blind man. You can perceive vibrations, and you have a direct measurement of the structure of the matter at the atomic or quasi-atomic level, or you can detect electric charges. So one end of this AC circuit, you can see the resistances here, is made by this sandwich. The microtubule growing on this pad of nanoelectrodes, and the other part of the circuit is the tip of the atomic force microscope touching the same microtubulus, and you can see a nice voltage to current linear relationships. What is amazing is that he also created direct circuits with 4 gold nanoelectrodes on a single growing microtubuli, and by comparing the DC and AC resistance of these circuits, he found out that the current was changing stepwise. This is a clear indication of a memory system. So he applied to a single neural cell microtubulus the usual code that we may have in a computer: write, read, erase, and write again. And for each cycle of applied inputs after an erase, the conductivity of the system was starting from a higher level. That means that the system has a memory. And the latency for such a switch was almost close to 0. This is what we call hysteresis loss of the system. So that means that no human computer is still able to do this. We have this inside a single microtubuli. Then he went further. He published this paper, I don't know if you can see, it's Nature Scientific Reports. Scientific Reports is one of the newborn journal in Nature family that only deals with seminal discoveries. He was able, Enerman was able to afford a live visualization of single isolated tubulin protein self-assembly via tunneling current. What does that mean? That at the end of the day, he constructed an artificial cell of 500 microns diameter, roughly, with electrodes that can pump AC, alternate current signal, into tubulin molecules that are assembling to form a microtubulus inside this nanochip. And with the coupling of scanning and tunneling microscopy and atomic force microscopy, it was able to come up with this amazing images. You can see now, these are the electromagnetic resonance pattern from a single tubulin molecule, the building block for this microtubuli. These are megahertz, the symbol M, so no signal. When 37 megahertz are applied, you can see this, you can visualize the structure of the conductivity of the building block of the single microtubuli, which I'll show you later is essential for our cell signaling. Without this, we won't have any cell signaling. We don't have any patterning in our cells. So whenever you change the frequency, 160, 150 also works. That way, Dieter knows why I'm saying this. You can see the dramatic change in the molecular structure, but this is a structure that is the conductivity structure of the system. When you go to the microtubuli, once again, remember, I'll show you this structure are the major cable through which the information goes inside the cells. Changing the electromagnetic field that you're pumping inside this microtubuli will change dramatically the structure of the microtubuli and its own conductivity. Of course, accept this for a moment. It may look very complex, but then at the end, I will try to make this picture very simple. And like having many dancers that at the end of the day, they start to dance together. And this is a deep meaning for the cellular biology. So you see, and each frequency has, in this case, 3 major resonance forms, magnetic forms for the microtubuli. You can see under your, this is an amazing, this is a seminal discovery. Now, can we use, so our cells are producing magnetic fields. The mechanical vibration of the microtubuli can produce magnetic fields. The conveyment of electromagnetic fields to these microtubuli within the megahertz range is able to afford changes in the structures of the proteins that last in the microsecond range, which is important for the microtubuli to stably change their structure and their function for the cell. But can we do the reverse? Can we use electromagnetic fields to change the fate, the commitment of our cells, or better, our stem cells? This was the first evidence that we published in FASEB journal in 2004, but we started with this experiment in the late 90s. So we can turn on cardiogenesis with extremely low frequency magnetic field. In those years, we discovered the master gene for cardiogenesis. It's dynorphin. It's an endogenous opioid peptide. It is an endorphin peptide that was thought to be produced only by the brain. At those times, the heart was only a mechanical pump. My project at the NIH was to demonstrate that the heart is like an endocrine gland and can produce the same substances that are essential for the nervous system, the central and the peripheral nervous system dynamics. And we did. Dynorphin B is crucial for the brain function. But as you can see here, these are mouse embryonic stem cells. Usually, they spontaneously start to contract, but the yield, the final yield of myocardial cells is only 0.002%, so it's very low. This is also happening in a human being, because an embryonic stem cell doesn't have to make only the heart, it's to make the entire body. So the yield of cardiogenesis is extremely low, like the yield of neurogenesis. And in order to take advantage of stem cells for regenerative purposes, we had to increase by several orders of magnitude the strength of this cardiogenesis. And we succeeded with chemistry. You can see each single dot spot here is a single cell. So we transformed the very low efficiency event, cardiogenesis, with chemistry, with an endorphin peptide, in a very high throughput outcome. This was the first evidence in literature that we can create a beating microheart in vitro. This is the effect of a magnetic field of 50 hertz, 1.8 millitesla. So it's the same magnetic field that goes around the power supply, the electric domestic power supply. Same story. Many cells, they come together, they aggregate, and form a microheart in vitro, synchronously beating. Then, this is just to be rigorous. All the things that you've seen, they pass it through the effect of the magnetic field to specifically enhance these guys, which are transcription factors we call cardiogenic transcription factors. These are molecules that go inside the DNA, and they trigger specifically a commitment of the cells, the stem cells, toward the cardiac lineage instead of any other lineage. So it's a quite specific signature. We made all the experiments at the transcription level to prove that that was true, and even if you look at the confocal microscope, the expression of cardiac-specific protein and untreated cells and cells that had been exposed for 7 days to the magnetic field, you see the clear-cut difference. Each dot spot is a cell, and the green light is a contractile protein that is specific for the cardiac myocytes and make these cells contracting, as you've seen before. Now, just to reinforce the relevance of this finding, we have seen before brain and heart. So the endogenous opioid peptides are crucial for the myocardial function, but at the beginning they were believed just to act on the brain. And not only the heart is a target, but it is also a source for these endorphin peptides. We published in very rigorous journals like Journal of Biological Chemistry. And in the official organ of the American Heart Association in 2000, Circulation Research, we published this finding that this endorphinergic system is able to transform a mouse embryonal carcinoma, the most powerful cancer cell for rodents, into beating myocardial cell. So this is official. I wonder why this knowledge has not been deployed and exploited further, because this could have changed many things. And it was published in a very official journal. Here, it's all, you can see what we have published in detail. But based upon this experience, we wanted to go to human adult stem cells and not only treat these stem cells with a new device in vitro, but based upon the diffusing feature of a magnetic field or a sound vibration, we'll see later on, we wanted to target with the stem cells where they are, without the need for tissue or cell transplantation. So this is a new strategy in regenerative medicine, relying upon the cell, decreasing the threshold for our self-healing potential. That's the main point. And this is the new guide that we developed. It's called REAC, Radio-Electric Asymmetric Conveyor. How does this work? It's apparently a regular emitter. It emits at 2.4 gigahertz. You know that your internet card in the computer is working at that frequency. There is nothing specific for that frequency. Okay, Dieter may say it's the 16th harmonic of 150 megahertz. This is true. But we choose that frequency simply because we didn't want to have trouble with regulatory agencies. And because the mechanism that we had in mind was based upon a completely different idea. Was not necessarily the frequency touching the cell, but the innermost response to the cell that we have to pick up and give back to the cell to make the cells aware of what they were doing in response to a very weak magnetic field. So I will try to convince you that consciousness is expressed at any level not only the entire human being, the universe, but the cell, the molecular, and you have heard electron, even at the lower level of dimensions, we may find this connectedness. So it's well known that whenever a living tissue interacts with a magnetic field, that tissue, the cells in that tissue will develop a microcurrent. This is well known. So we decreased the strength, the radiated power, the power of the emitted field up to 2 milliwatts. The card, the Wi-Fi card for internet connection in computer is about 10 to 12 milliwatts. And since we emit like a black body, our radiation, our radiation has an intensity of about 58 to 60 milliwatts. So this machine emits a very weak electromagnetic field. Nevertheless, it will trigger a microcurrent in the body, in the whole body. Now, the peculiarity of the system is that we invented this probe. This probe picks the microcurrent that you're generating, picks the microcurrent up and sends back to the area of the body that you would like to trick. So the sample holding and release time, the pulse time, is 250 milliseconds. And the interval between pulses varies between 2.5 and 5 seconds. So in this way, you're picking up mainly the endogenous response of your cells to the magnetic field. And these cells will generate more and more current, so the system has no depth limits. You can focus the area of the body that you would like to treat. The underneath tissue will generate a resultant microcurrent. This resultant microcurrent will be conveyed back to the tissue. This generates the molecular, biochemical, and later on you will see clinical response. So start to have this term in mind, polarity, cell polarity. Without polarity, our cells are dead. Our cells are not symmetric. A healthy cell is not symmetric. The apex of a healthy cell is different from the basal area. The lateral parts are different. They do vibrate with a specific direction. You have seen this. So there is mechanical motion with a targeted direction, and there is a field that goes in specific direction. Whenever the cell is symmetric, I'll show you many other papers from other groups, not only our group, even published in Nature and Science, that prove that a perfectly symmetric cell is a strong cancer cell. So, yeah, yes, really. So we developed this system. The probe, this is inside the incubator. This is the REAC device, an old one. We have a new one built up recently. The emitting radiating antenna, and the probe now, you see, it goes inside the tissue culture dishes. This is a laminar probe that picks from the stem cells the microcurrents up and give them back to the cells. So the cells are excited by the emitted magnetic field. They produce, in response to the field, an endogenous microcurrent. This microcurrent is given back and back to the cells until they optimize their polarity. And the result of this is that we can handle as we want, as we desire, the differentiating potential of embryonic stem cells. They can become neural cells, skeletal muscle cells, myocardial cells. The yield of myocardial cells developing with this method is well evident. Control non-exposed, exposed cells. If you just pick the few cells, if you pick the few cells up that do spontaneously undergo cardiogenesis, you can see here, this is what a network of the few cells that become spontaneously cardiac myocyte looks like. But here's what we found after REAC treatment. You see, you can see this area here, which is beating before, and then a waveform loop of contraction goes around. So primer stimulus, contraction, primer, contraction. This is a pacemaker. The first time that we were able to transform, in this case, mice stem cells, in pacemaker cells, and then in heart. So it's completing the structure of the heart. But we wanted to go to clinical practice. So we moved to human adult mesenchymal stem cells, isolated from the adipose tissue. We placed these cells in culture. To cut a long story shorter, we exposed the cells for 48 hours to react to this treatment, and then we left the cells untreated up to a total period of 10 days. You can see all these names here, prodynorphin, whatever. These are genes for the cardiogenesis. These are genes for the vasculogenesis, for the formation of new vessels, which are very important. Just imagine it, coronary vessels disease, the stroke, diabetic, vascular diseases, and so on and so forth. We induced neurogenin 1, which is the master gene for neurogenesis. MyoD is the master gene for skeletal myogenesis. Now, probably you remember in 2012, Shinya Yamanaka and John Gordon, they got the Nobel Prize for reprogramming human adult mesenchymal stem cells, or even somatic human non-stem cells, like somatic cells of the skin fibroblasts, into embryonic-like stem cells. There was a seminal discovery. That means that our cells are not targeted for their future. They can go back to the ancestral memory, as I said before. They can retain the memory from the fertilized oocyte, and they can recall this memory, provided that you give them the right signal. Unfortunately, what Yamanaka and Gordon did is using viral vectors. It's called lentiviruses. A lentivirus is a modified HIV vector that contains the genes that you would like to introduce into the cell, and you would like to introduce inside the DNA. Makes sort of scar into the DNA, and then the DNA is able to express these genes that are supposed to be active only in the embryonic phase of our life. They succeeded. They made even dermal skin fibroblast becoming cardiac neural cell, but the yield was very poor, less than 0.1%. The remaining 99 point something percent were undifferentiated cells with a very high risk for tumor. Plus, they've been using a viral vector, which is a modified HIV virus. So their discovery, it is a seminal discovery. It deserves a Nobel Prize, but cannot go into clinical practice. And there is one reason why the yield was so poor. Because if you introduce a gene with a vector inside the cell, that gene will remain activated forever. Now, it's a matter of timing. It's a matter of chronobiology. These pluripotency genes, and they have a name, SOX2, OCT4, NANOG, KLF4, they are not activated forever in the embryo. They are activated just for a few days, 2 or 3 days in the early embryogenesis, and then the cells become able to be committed to everything. But in order to complete their differentiation, these genes need to be shut down. We know why. It's a very long explanation. We'll take the entire lecture, but we know why we need to shut this gene down. But if you use a vector, you cannot do this. And as I told you, we treated the cells for 48 hours with this new way of delivering, conveying a magnetic field. And you see, first we get an upregulation of this gene on your right. During the stimulation, which lasted 48 hours, still during the stimulation, we had a biphasic response, an inhibition of these genes. So this later on made this picture come real. Human adult mesenchymal stem cells, which are not able per se to develop into cardiac, neural, skeletal muscle, vascular cells, can transform with a high yield efficiency in all these cell types. So by pre-treating ex vivo these cells with this form of magnetic field, we can afford a high efficiency program for cardiogenesis, neurogenesis, vasculogenesis, skeletal myogenesis. But this was not our terminal goal. We just wanted to demonstrate that this field is able to transform human adult cell. And we went further. And when we made this discovery, Yamanaka sent an email to us. You did it. What we did, we transformed human dermal skin fibroblasts into cardiac, neural, skeletal muscle cells with physical energy without using any viral vector. And you can see these are all the genes that Yamanaka and Gordo introduced with a viral vector in human skin fibroblasts. We triggered these genes on only with a properly conveyed magnetic field, but the effect was biphasic. During the stimulation, first an overexpression of the genes, look on your right side of the picture, and then still during the stimulation, a downregulation. This was the final outcome. These are cardiac cells, skeletal muscle cells, neural cells coming from skin, adult, somatic, non-stem cells of the human body. And then we went further. As I told you, a major problem is aging of our stem cell population. With this approach, we were able not to prevent, to reverse stem cell aging. And this paper was published in 2014 in Nature, Scientific Reports. So let me show you. These are all pictures that show that we were able to reverse stem cell aging. The expression of senescence-associated beta-galactosidase went down. All these genes that are related to the ability of the cells to become pluripotent. BMI1 is a master gene that triggers the activity of the stemness genes that I've shown you before. So it's sitting on the top of this hierarchy scale. And we grew the cells for 30 passages, which means 3 months. So these are aged cells by definition. They are not able to do anything. Whether they are somatic or stem cells, they're not able to do anything. It's a well-consolidated model to destroy cellular activity through an aging process. We reversed the gene transcription from an almost dead state to a very active state. You see all the pluripotency genes went back. Nanog, CMYC, these cells started to proliferate again. And the reviewer asked us, but then you have a risk to induce a cancer. What happens if you keep going and going? Then the cell all die. That means that we are not creating a model for immortalizing stem cells, which is maybe the real danger for this treatment. Okay, this is important. TERT. TERT is a gene which encodes for the catalytic core of telomerase. So we were able to re-elongate the telomeres, which are the major point where the aging, non-aging decision is planned in these cells. Here, sorry. Okay. PC12, cancer stem cells. This is what I told you. Another paper in Nature. And we were able to reprogram a cancer stem cell into a normal stem cell, up to the point that after REAC exposure, we were able to re-express beta-3 tubulin, NGF, and tyrosine hydroxylase, which are markers for neurogenesis, with specific differentiation into dopaminergic neurons. This was true not only at the gene, but even at the protein expression level. You can see in the absence of treatment, the expression of these neurogenic markers is very low. It dramatically increases after the treatment. The expression of tyrosine hydroxylase, which is the hallmark for then producing dopamine, is dramatically increased. We have used these neurons to treat Parkinson's disease in an animal model in mice successfully. Then cell polarity. It's another paper that we published in Nature recently, right now in June. And we asked us, what's the mechanism? Can we abolish this ability of this technology to reverse stem cell aging? Because that means that not only we understand that it goes through gene expression level, but we may understand some inherent features, some deeper mechanisms behind this. And the answer is yes. We use the molecule, this one, 4-MU, which is inhibitor of type 2 hyaluronan synthase. Now, this is the major enzyme that gives rise to the synthesis of hyaluronic acid. Hyaluronic acid is a major component of extracellular and intracellular matrix. So, as I said, okay, we are made of water, but not as a salt solution. This is a major mistake that has been done throughout all these years. We are made of water in terms of non-homogeneous gel. And as I show you in a moment, signaling molecules cannot swim in this gel. So the old model of a lock and the key is not working anymore. It cannot explain what you experience every day. If you meet somebody that means something for you, or you listen to music, you feel your body is changing. It's impossible that these pure molecules are traveling all over the tissue, swimming, looking to each other. It takes too much time. There is another mechanism that I'll tell you in a moment. But just to say that we know the inherent mechanism that we are producing with this field is optimizing cell polarity at the mechanical, structural, vibrational, electrical, and magnetic level. Now, this is also why, despite all the time that they tried to stop us, this device is finally a medical device acknowledged by the European community for cardiovascular stimulation aimed at reparative processes and electrophysiological optimization of the heart, or neurostimulation and biostimulation. But the sad part of the story that we don't have enough money to develop this. So it's a prototype. We're going into clinical practice. We can do clinical trials. We can do observational trials without any permission, or we need the ethical committee if we want to do large clinical trials, which we are actually going through. But producing this will become a disruptive technology of the status quo. So anyway, this is another issue. Do I have 10 more minutes? Yes. Sure? Okay. Okay. So, because I want to conclude the picture and try to show you the meaning of the first part, the vibration, and try to make it clear why it is connected with the magnetic field. So, we can reprogram the cells with this sound, and the sound can also produce not only mechanical but electromagnetic vibration. That's the point that I'll try to address in this last few minutes. So, first of all, imagine this microtubuli that I've shown you at the beginning, the vibrating stuff in the cell, like strings, like road, and you can imagine the signaling molecules, whatever, enzyme, transcription factors, as an oscillator. This parallelism is more than accurate, because every single signaling molecule, if I can, works. Okay. It's alpha helix, a turn, alpha helix, turn, alpha helix, so on and so forth. So the major building block is very simple. This is like a spring. This is a connector between springs. If I touch here, I make resonance mode. So the mechanical input here is non-local by definition, but I will have lysine, arginine, histidine, aspartate, glutamate, positive, negative charges. I make a physical vibration of a system that will induce electromagnetic fields. And magnetic fields within the megahertz range will make the system vibrating. So the two things they can be mutually coexisting. Now, if a signaling molecule is an actuator, is a vibrational actuator, mechanical, magnetic, then the question is, how do these oscillators recognize each other? The classical picture was, one is the lock and the other is the key, but it's too slow. Plus, there is no bulk water in the cells. This is proven. There is no free water. It's all bound to surfaces. And the chance for these poor molecules to swim is very low. And the cells are made at very high content of hyaluronic acid and other glycosaminoglycans, so molecules cannot simply swim. They must walk through these cables. And let's try to understand why and how. So each metronome here is like a signaling molecule. It's like that. It's an oscillator. Is it clear? Okay. Each string that makes a row of metronomes, each metronome's row is like a cable. Each row is connected to the other row by an elastic spring that you cannot see. So if I put these metronomes onto a rigid surface and they start non-synchronous, they will never come to synchronicity. It will take forever. But if I do this in this situation, where each elastic system dissipates the differences in vibration, you see, they are not synchronous. Is it a way that I can make the movie going up to the end just to save a couple of minutes?
You can speak while they continue to move.
Yeah, okay. If you just go down, perfect. You go right here, okay, maybe here, okay. Okay, that's enough. In a few minutes, they become synchronous. It's not a joke, because this is the supplementary movie of a nice paper that appeared in PNAS, a major journal, which tells us that all over around the universe, whenever we had a vibrational entity, and this vibrational entity is placed upon an elastic surface that dissipates differences in vibrational patterning, the system goes through a chimera state. What does it mean? It's very democratic. I can imagine that synchronicity can be achieved because some god tells, oh, you have to dance at this frequency, and tell you what the frequency is. But this is a spontaneous behavior. Chimera means that, you can see here, for a certain period, the metronomes oscillate in a apparently chaotic, which means scale-free mode, and that means that their frequency and their rhythm is not the rhythm that each individual oscillator had before the experiment, but it's not the final rhythm. They find the final rhythm each other by sharing optimized vibrational singular individuality. So that means that the final result is not an imposition. It's the result of individual behavior that has the inherent capability to perform as a collective behavior. So, the main message here is that we're not losing our individuality if we try to simply respect what nature is, connectedness. If we think to maintain our individuality, that's simply maintaining our negative ego. So, the metronomes are our proteins. Now, let's try to give an example of consciousness and connectedness at the molecular level. This paper came out in nature, nature communications. So these proteins, helix, turn helix again, helix, loop helix, are the major signaling proteins. Even the DNA, in order to stay in such a tiny space, has to build up such a configuration beyond the double helix. Now, this is like the metronome. Can you see a spider on a web? So the spider is like the metronome. It's like the vibrational entity. It's like the signaling molecule. And the web made by the spider is like the network of microtubule. Is it okay for you? Now, whenever a singular vibrational entity starts to move in a web, makes it exhibit connectedness. You want to call it consciousness? It's the same. That means that in the same web network, you may have a spider, a fly, a bee, a bigger bee. They are like oscillators. They will never find each other if you put on a rigid surface. But on this system, the system will dissipate the differences in the vibrations, and maybe not a fly will recognize a big bee, but a big bee can be recognized by a larger fly. And how do they recognize to each other? They start to oscillate in synchronous. And what happens on the web? If there is another vibrational entity here, you will see a string coming out in this web. That string is a wave that travels across the web and informs each point of the wave, or the web, that something is going on. The two guys are connected. And this connectedness cannot be separated by the way the other guys not are previously connected, but then the way they will be connected after this event. So it's a way to manipulate space and time in 3 dimensions, as you see. And physicists know very well that the speed of phase velocity is the square of c, right? So it can approach synchronicity, which is not violating the speed of a photon. It's a different story. You're breaking the symmetry of the system. Breaking this symmetry, it's a new wave, it's a new phase wave that will travel at such high speed. And we have this in ourselves. Now, in this example here, the vibrational entity, like the spider web, are trapped in the web. They are not free to move. In reality of ourselves, we have these guys. This was published in Biophysical Journal, which is Science, actually. We had these molecular motors, kinesins. They hydrolyze ATP. They produce energy. They use this energy to walk and to walk onto this microtubuli. So the microtubuli, they do vibrate. They dissipate differences in frequencies of vibrations. The vibrational entity do not sit directly on this microtubuli, but they are on the shoulder of this man walking. This is called kinesin or dyne. So you can imagine that the vibrational entity is not here, but here. So it can dance on the microtubule, but it can move along. Then this is not contradicting the original old model, Jacobi-Monod, the lock and the key. That can still exist, but is a result of the vibration of each entity. So if I may have two entities that are so different in their vibration, they will never recognize each other in the mode that I described before, but by moving, touching each other, they may have some compatibility. And from two entities, that will become a new, single, transient vibrational entity in that point, in that moment, in that space, in that time of the cell. And all the other guys will know it. Is it true? Yes, because we can see. With the atomic force microscopy, we can see the microtubuli. You can see the tubulin molecule. You can see the feet of the molecular motor. You can see it moving along the microtubuli. Now, I told you, Yes, arts and science. And this, she's Masha Dimitri. She was a wonderful acrobat. And she was walking the line. Now, with her permission, I will say this is, can I have just the audio, maybe the cable? It was useful for the last slides. So she's like the kinesin, and this umbrella is like the signaling molecule, the metronomes that you've seen. Shows she's walking. The cable is sensing is walking. She's trying to reach another guy, another molecular motor with another kinesin. The first approach fails. It's a matter of time, rhythm, music in this. But the second one is successful. So connectedness is an evolving form of recognition. Now she has no signaling molecule, but still induces signaling by moving. Yeah. And nice conventional journal, electroacoustic behavior of the mitotic spindle. Okay. +1. Cell mechanosensitivity to extremely low magnitude signals is enabled by linked. Link is the linker between nucleoskeleton and the cytoskeleton. We're talking about piconewton range forces, so very subtle energy. If it is too strong, this link system is a kind of distortion and does not transmit the stimulus into the nucleus, because transmitting this mechanical forces into the nucleus means changing the structure of chromatin, changing the structure of the gene expression. Once again, we can have just a Band-Aid containing carbon nanotubes. These nanotubes vibrate at the temperature of the skin. They excite the deep muscle around the spine, and this avoids muscle failure and prevents people with Parkinson's disease from falling down and break their bones. So, nanobiotechnologies. Now, yes, I need the sound again. This is the demonstration that the sound is emitted at single cell level. This was the first demonstration we made, James Ginczewski, University of California, Los Angeles, and myself, probing with the tip of an atomic force microscope single yeast cells. Then we made the same experiment with stem cells, embryonic stem cells. We hope we can do more in the future together. And now the yeast cells are well known to change their metabolism according to the substrate they have to metabolize. And we found with the tip of the microscope that the vibrational patterning was changing according to the substrate type that they were using. We thought that an artifact at the beginning. Then we used mutant strains which were not able to metabolize a given substrate, and we noticed a change in the harmonic patterning of the vibration. Then it's well known that by changing the temperature in the presence of a given substrate, the way the yeast genes work is changing. The velocity, the transcription rate of this gene is changing. And now you can hear the sound from a single yeast cell changing the temperature. Okay. At 22 centigrades, at 26. So it's a sound of a single cell. At 30, it's a sound, because if I kill the cell with the tip of the microscope, it's just a background noise. So we came up with this. Ginczewski was part of the team. He didn't win the Nobel Prize because he was the fourth guy in the team. Richard Smalley won the Nobel Prize, but he won twice the Richard Feynman Award in Physics, which is almost a Nobel Prize. And we made this patent together that is, we can retrieve vibrational signatures. We call it sonocytology from the cells according to their healthy, non-healthy conditions, or if it is a stem cell, according to the way the cells is going to. Is it becoming cardiac, neural, whatever. And we are proving that the reverse is also true. We can record the sound of, let's say, cardiogenesis, neural differentiation, as a signature that we can give back to a non-differentiated stem cell to commit these cells to the same lineage. And once again, we made this chair. It's a vibrating chair which is delivering a specific sound using the spine of the patient as a second and resonant actuator. And we can actually use codes like feline purring, because you know that domestic cat, but even a tiger, they can heal themselves with purring, which is completely, it's a kind of purring completely different from the purring they make when they're happy. But there's also differences from the purring in the domestic cat and the tiger, except for the fact that if you do a spectroscopic analysis of the frequencies and you look for autosimilarity curves, you plot logarithm of the power of the sound versus logarithm of the frequency, you make a map with some fractal frequencies that are exactly the same in a tiger and a domestic cat. And we can use different protocols to heal internal organs or skeletal muscle or bones. This also applies to Parkinson's disease, tremor. It's a way to make the physical body even perceiving the music, if you want. And indeed, this is a crucial experiment. Cardiac transplantation in rat, the engraftment of the transplanted heart only lasts less than 10 days spontaneously, but it can last 3 months, which is consistent part of the lifespan of the rodent, if you use music, classical music or some form of jazz music. Unfortunately, some form of new age music didn't work, but I'm sorry for that. But classical music and jazz, they work very, very well. Now, I'm coming to the conclusion, cell polarity. I'm lucky that other guys discovered this already, so it's too self-referential. This other guys, cell polarity in cancer, cell and tissue polarity as a non-canonical tumor suppressor, commentary, Journal of Cell Science, Nature Cell Biology, cell polarity in the development and cancer. So there is a subtle line between normal development and cancer. Cancer is simply deviation from normal development. How this and why, how this deviation may occur, we may know. Why it pertains to non-scientific realm, and it's more deep meaning. Once again, epithelial cell polarity, stem cells and cancer, nature again. Cell polarity proteins in breast cancer. Nature communication, loss of polarity proteins promotes pancreatic cancer metastasis. So, take home messages. A new vision of the cell world and regenerative medicine. Body regeneration. We regenerate most of our body approximately every 3 months. So our body has inherent regeneration potential. We are naturally targeted for self-healing. There is a population of resident stem cells that can drive a regeneration process that is distributed across all the entire body. Each individual is a small piece of nature. We live in a multitude of physical signals and fields. The human body is incisively penetrated and crossed by a number of cosmic particles, energy waves, electrically charged or not. Once again, we are part of nature. Our body generates seeming infinity of rhythms and vibrations expressing the cell's molecular dynamics, now recordable at the atomic and molecular levels. Different cells may have different languages. If you record a culture of healthy cell, old cells, cancer cell, you can capture different languages. Through these reciprocal physical signals, the cells are able to organize their decision. We live in an active physical environment. The body cells Both generate microcurrents and are sensitive to external physical fields and acoustical subsonic vibration, and can react with profound functional changes, including transdifferentiation, until clocking back to a time zero in which all kinds of decisions are still possible. Ultimate goal, to reprogram the tissue-resident stem cells with no need for stem or cell tissue transplantation by a non-invasive, inexpensive, personalized, and easily deliverable on large-scale basis technique. A person does not hear sound only through the ears. He hears sound through every pore of his body. It permeates the entire being. According to the sound and its influence, a certain effect is produced. Sound becomes visible in the form of radiance. This shows that the same energy which goes into the form of sound before being visible is absorbed by the physical body. In that way, the physical body recuperates and becomes charged with new magnetism. My brain is only a receiver. Nikola Tesla. They called me crazy. Now, soon this history will repeat itself when I say that I have discovered a previously unknown source for energy, boundless energy, which can be funneled. And this we wrote with Jen Jinczewski and Richard Smalley. In both philosophical and visual sense, seeing is believing does not apply to the emerging new discovery in science, for which there is nothing even remotely visible to create a proof of existence. On the atomic and molecular scale, data are recorded by sensing and probing in a very abstract manner, which requires complex and approximate interpretations. We are now facing a novel path where visualization and creation of a narrative becomes necessary to describe what is sensed, not seen. We have growing needs of separating the information content of life from its material substance, information, and deploying it into multifaceted knowledge are the essence of life. Now I can tell you what this symbol is. Stem Wave Institute for Tissue Healing. This S is like a stem, but it's also like a healing entity, which is drinking water from a wave, water. And this picture here, which is being made by Michel Ferry, an artist who lives in France, a friend of us, as a whole. This is where we are. We hope this all will bring us to a new dimension. I want to thank all the people working with me. Emilio Del Giudice was a wonderful quantum physicist who died 2 years ago. And thank you very much.